We are working in the eye group, and why we are working in the eye, especially we are working to trying to find a cure for macular degeneration, and we are focused in cells that are beneath the retina. These cells are retinal pigment epithelium, and these pigment epithelium cells, since evolution, they have the property to regenerate retina in lower vertebrates like frogs, salamanders, and for some reason in higher vertebrates these cells are like dormant. But we find that when you take those cells out of the eye, these cells start to present some plasticity.
Derek Vanderkoy. I'm from the University of Toronto in Canada.
Okay, and what type of research are you doing?
Lately we've been working on stem cells in the eye, in particular the back of the eye, the retina, and we've discovered that stem cells are present in the edge of the eye in adults and in embryos. And the retinal stem cells are the only cell you can actually see without cutting into the body. So if you look deeply into my eyes, you can see the colored region, which is the blue or the green or the brown region, and that's the iris. And if you look just outside of that, there's a very thin black line that surrounds the colored region, and that's actually where the retinal stem cells are. There are 1 in 500 of those cells, and if you take them out of the adult eye, even from a person that dies when they're 80 years old, one of those cells can proliferate in a week to produce 10,000 cells. Those cells will become the different types of cells in the retina. They'll differentiate into photoreceptors and amacrine cells and bipolars and retinal ganglion cells. And so one of the things we're trying to ask right now is, can we actually take a human retinal stem cell from an adult human who's died, grow those cells up in culture, make large numbers, and then put them back into mice that are blind because they have a mutation that makes their photoreceptors not work correctly. And the question is, can a human photoreceptor connect up with the retina of a mouse that doesn't have any working photoreceptors and give vision back to a blind mouse?
Hello, I'm András Dínyes from Hungary. I'm representing Biotalentum Limited, which is located at Gödöllő, close to Budapest. And our research is focused on embryonic stem cell and iPS research at the moment. We used to do nuclear transfer cloning, but as the field is moving very quickly, it's really nice to see that reprogramming and pluripotency is coming up with new potential for human medicine. There is a need for standardization and modeling. So one of my concerns actually that mouse modeling is running well, but there is a big gap between the mouse and the human. And for the applications there is a need to develop more probably large animal models, primates, but primates are not very easy, so I have a strong interest in developing rabbit, bovine, pig models for testing the systems and the regenerative medical applications.